In this topic, we're going to discuss the immune system, B lymphocytes, and humoral immunity. So we're going to look at the cells of the immune system, features of lymphocytes, then maturation of B lymphocytes, response to antigens, which is called the humoral immunity related to B lymphocytes, plasma cells and memory cells, a graph of the primary and secondary immune response, the role of B lymphocytes in the immune system, plasma cells and how their structure relates to their function. Now, the last topic we discussed the cells of the immune system. Can you remember what they are? Well, you've got your phagocytes and lymphocytes. So today we're going to look at the B lymphocytes. Your lymphocytes comprise B lymphocytes and T lymphocytes. The B lymphocytes are involved in humoral immunity, so they secrete antibodies. Your T lymphocytes are involved in cell-mediated immunity. The features of lymphocytes are they are smaller than phagocytes and they comprise B and T cells, or B and T lymphocytes. They've got a large nucleus. So if you look in this diagram here, you can see the lymphocyte has got that large nucleus that dominates the cell. Now, lymphocytes are produced before birth in the stem cells of the bone marrow, and then they circulate in the blood and the lymph. So let's just have a look at the overview of the two types of lymphocytes, and then we're going to discuss B cells in more detail. So both B and T cells are made in the bone marrow. The B cells remain in the bone marrow until they are mature, whilst the T cells leave the bone marrow and then they mature in the thymus. The B cells circulate in the blood and then they concentrate in the spleen and lymph nodes. So their names indicate where they develop. So B lymphocytes, or B cells, mature in the bone marrow, and T lymphocytes mature in the thymus gland. So here you can see how the B lymphocytes develop. They're made in the bone marrow of the fetus before birth. Then maturation begins after birth. They then circulate in the blood and lymph and concentrate in the spleen and lymph nodes. So looking at this diagram, you can see that the immature B cells are made before birth. Then the maturation begins after birth, and each cell gains the ability to make one type of antibody. This is under genetic control. These cells now divide by mitosis to form new clones, and each has the ability to make a particular protein. Antibodies in the cells form the protein receptors, which are displayed on the cell surface. And these receptors are what combine with specific antigens. So if the antigen is present, the cell surface receptor will recognize it, and then your B cell is going to produce antibodies. So these mature B cells will move to the blood and concentrate in the spleen and liver. Now we saw in the last topic how when a body is invaded by foreign material, this material possesses antigens that stimulate the B lymphocytes to produce antibodies. These antibodies are soluble in the blood and tissue fluid of the body. So another word for body fluid is humor, and hence the production of antibodies is known as humoral immunity. You've got lots and lots of different types of B lymphocytes, and each type produces a different antibody which responds to one specific antigen. So in this diagram here, you can see the antigen binds to the B cell receptor. The B cell then becomes a plasma cell which secretes antibodies. Okay, looking at the different steps in the humoral immunity. A pathogen has got surface proteins that we call antigens. So here you can see the pathogen in green. It is these that the cell surface receptors of the B cell recognize, and a primary response is initiated whereby the B cells will divide repeatedly by mitosis, forming identical clones. So this is called clonal selection. Some of the daughter cells can become plasma cells on the right, and others will become memory cells on the left. The plasma cells have got extra endoplasmic reticulum, and they synthesize antibodies. These antibodies are specific to the antigen, and they bind to the antigen on the pathogen. The antibodies can destroy the pathogen or the toxins that the pathogen secretes. The other daughter cells that have become memory cells have got long-term immunity, and they remain in the circulation of blood and lymph. These memory cells form immunological memory, so that if the same pathogen invades again at a later stage, they're going to initiate a secondary response. And this secondary response is much faster than the primary immune response. And the memory cells are able to divide to form plasma cells that secrete antibodies, and this process is much faster. So I mentioned that B cells divide to form plasma cells and memory cells. The plasma cells secrete antibodies. They usually only survive for a few days, but each cell can make around 2,000 antibodies every second during its brief lifespan. So these antibodies destroy pathogens and any toxins that the pathogen produces. The plasma cells are therefore responsible for the immediate defense of the body against infection. So this is known as the primary immune response. The memory cells live considerably longer than plasma cells, often for decades. These memory cells do not produce antibodies directly, but rather they circulate in the blood and tissue fluid until they come into contact with the same antigen at a further date. So when they do so, they rapidly divide and develop into plasma cells and more memory cells. And the plasma cells produce the antibodies and secrete them so that the pathogens are destroyed. And then your new memory cells circulate in readiness for a further infection. So looking at this graph here, you can see that the secondary immune response is much faster and it's larger. So the memory cells provide long-term immunity against the original infection, and this is known as the secondary immune response. It's both more rapid and of greater intensity than the primary immune response, and it ensures that the new infection is repulsed before it can cause any damage. So notice in this graph you've got antibody concentration on the left and time at the bottom. How when a person is first infected with the antigen, the primary immune response is activated. You can see that there are a few antibodies secreted in response. Now have a look at the secondary immune response with the same antigen that infects the body later. Can you see how much higher the curve is and how quickly the secondary response is activated? Why? This is because the body has got memory cells that identify the antigen, and they can quickly divide to form plasma cells that secrete the antibodies. Okay, let's have a look at the role of B lymphocytes in the immune system or the immune response. So here's a lovely diagram to show you the role of the B lymphocytes. The invading pathogen produces antigens that are taken up by both the B lymphocytes and macrophage cells. So you can't see the macrophage cells in this diagram here. The B lymphocytes process these antigens and then they bind them to a protein called the MHC, which stands for Major Histocompatibility Complex protein. The MHC protein presents the processed antigen on the surface of the cells, which are then referred to as antigen-presenting cells. So here's another sweet diagram to show how a phagocyte engulfs the bacteria and digests it, presenting the antigen on its cell surface. The T helper cell in purple there attaches to the processed antigen on the macrophage and becomes activated, making it capable of interacting with B lymphocytes. So in this diagram here, the T helper cells attach to the MHC proteins with the antigens on the surface of the B lymphocyte. The cytokines are secreted by the T cell, and these help the B cells to multiply and mature into antibody-producing plasma cells. So the B lymphocyte is activated to divide by mitosis to give a clone of plasma cells. The cloned plasma cells produce antibodies that exactly fit antigens on the pathogen's surface. So the antibodies can attach to the antigens on the pathogen, causing agglutination and then lysis of the pathogen so that the pathogen is destroyed, and we call this the primary response. Now, some plasma cells develop into memory cells that survive for long periods, and future invasions by the same pathogen lead to rapid division of memory cells, some of which develop into plasma cells that produce antibodies. So this is called the secondary response. Right, plasma cells differ from B lymphocytes, from which they develop in a number of different ways. These differences relate to plasma cells' function of producing antibodies at a phenomenal rate. So compared to B lymphocytes, they have a large size, and this is to accommodate many organelles needed to manufacture antibodies. They've got more mitochondria to provide the energy needed to make antibodies. They've got more rough endoplasmic reticulum. This is to make and transport the proteins that make up the antibodies. And they've got more Golgi apparatus. This is to sort, process, and compile the proteins into antibodies so that they can be secreted. So the structure related to the function you would need to know if you had to identify the different parts of a cell, a plasma cell, and discuss what their functions are. Right, the summary of the primary immune response. Okay, it's active immunity. So a primary immune response is initiated. B lymphocytes recognize the antigen and then they divide by mitosis. So this is called clonal selection. Some of the cells become plasma cells and others become memory cells. The B lymphocyte plasma cells synthesize antibodies, And these antibodies are specific to the antigen. The memory cells give long-term immunity so that you've got a faster secondary response. So the memory cells recognize the antigen and then they divide to form plasma cells that secrete antibodies. And involved in the process, you do have T helper cells that secrete cytokines, which enhance the humoral response. And that concludes our lesson. The end.